Hello, everyone. My name is Matt Wilderson, and today I will be taking you through an in-depth look at red blood cells, specifically Chapter 53 of Harper's Illustrated Biochemistry. We'll explore their structure, metabolism, life cycle, and connect all of this to clinical relevance. So, without further ado, let's get started. So, our learning objectives break down into 8 different parts. 1, overview of red blood cells. 2, red blood cell structure and membrane composition. 3, hemoglobin biochemistry. 4, erythropoiesis and life cycle. 5, energy metabolism in red blood cells. 6, clinical and lab parameters. 7, integrated case and example. And 8 is the summary. So, the section 1 overview of red blood cells. Well, red blood cells, or erythrocytes, are the most abundant cell type in the blood. Their main job is to transport oxygen from lungs to tissues and return carbon dioxide from tissues to the lungs for exhalation. They achieve this by packing themselves with hemoglobin and an enzyme called carbonic anhydrase, which helps convert CO2 into a more soluble form of bicarbonate for transport. They are tiny and mighty, making up roughly 40 to 45% of our blood volume. This huge presence ensures tissues to get the oxygen they need. When hemoglobin levels fall below about 120 to 130 grams per liter, we can call it anemia. And that can significantly reduce oxygen delivery, affecting energy levels, organ function, and overall health. So 2, red blood cell structure and membrane composition. One of the first things you notice about an RBC is its shape, a bioconcave disc. This isn't random. The shape increases surface area for gas exchange and allows cells to bend and squeeze through capillaries narrower than the cell itself. And as we see in the bottom right is an image of the top view and the side view. So the RBC membrane is composite of 3 main elements: a lipid bilayer for flexibility, integral membrane proteins, and peripheral proteins. So for integral membrane proteins like band3, which exchanges chloride and bicarbonate to acid CO2 transport, and glycophorin A, which carries many blood group antigens and even serves as an entry point for certain pathogens. For peripheral proteins like spectrin and actin and protein 4-1, which form the cytoskeleton, this network keeps the cell strong enough to maintain its shape, but flexible enough to deform under stress as well. These structures aren't just for stability, though. Mutations in them can lead to hereditary conditions like spherocytitis and epileptitis, where the cell loses its flexibility and gets destroyed prematurely in the spleen. Hemoglobin biochemistry. Inside the red blood cell is a dense concentration of hemoglobin, roughly 1/3 of the cell's weight. Hemoglobin is a tetramer made of 2 alpha and 2 beta chains, each holding a heme group with an iron atom in the ferrosis state, which is Fe2+. Oxygen binding is cooperative, meaning the binding of one oxygen molecule increases the likelihood that others will bind. This is why the oxygen dissociation curve is sigmoidal. The Bohr effect comes into play here, with lower pH or higher CO2 levels shift the curve to the right, releasing oxygen more readily to tissues. The molecule 2,3-BPG produced in red blood cells also lowers hemoglobin's oxygen affinity, helping unload oxygen in tissues. If the iron in hemoglobin is oxidized to ferric Fe3+, it forms methemoglobin, which cannot bind oxygen. Fortunately, red blood cells have NADH-dependent cytochrome B5 reductase system to reduce it back to its functional hemoglobin. Without this, methemoglobinemia can occur, causing cyanosis and impaired oxygen delivery. Erythropoiesis and the life cycle. Red blood cells originate in the bone marrow from the hematopoietic stem cells under the influence of cytokines and, most importantly, erythropoietin, or EPO. These stem cells commit to the erythroid lineage. This process is stem cells to protomonoblast to reticulocyte to the mature erythrocyte. Reticulocytes still have residual RNA and organelles, but within 24 hours they fully mature. A healthy adult produces about 2 million red blood cells every second to replace the less than 1% that are removed daily. Each cell lasts about 120 days before being cleared by macrophages in the spleen or liver. And EPO production ramps up when oxygen levels drop, such as high altitude or in chronic lung disease. Clinically, synthetic EPO is used to treat anemia and chronic kidney disease or after chemotherapy. Now, energy metabolism in red blood cells. Red blood cells lack mitochondria, so they can't use oxidative phosphorylation. Instead, they rely entirely on anaerobic glycolysis to produce ATP. Glucose enters the cell via GLUT1 transporters, which work independently of insulin. A small portion of glucose, about 5 to 10%, enters the pentose phosphate pathway, producing NADPH. And NADPH is vital for keeping glutathione in its reduced form, protecting the cell from oxidative damage. Red blood cells also have the 2,3-BPG SUN, which regulates hemoglobin's oxygen affinity by stabilizing its low oxygen T state, ensuring oxygen releases in the tissues. Clinical correlates and lab parameters. In the clinic, we evaluate red blood cells' health with a complete blood count. This includes red blood cell counts, hemoglobin concentration, hematocrit, and red cell indices like MCV, MCH, MCHC, and RDW. Different anemias present with characteristic lab patterns. Microcytic anemia from iron deficiency, macrocytic anemia from B12 or folate deficiency, and hemolytic anemia from enzyme deficiencies or membrane defects. On a blood smear, you might see Heinz bodies, denatured hemoglobin aggregates, or polychromasia, which indicates an increased number of reticulocytes. Integrated case example. Let's take G6PD deficiency as an example. This X-linked disorder affects the pentose phosphate pathway, meaning patients can't make enough NADPH to keep glutathione reduced. When exposed to oxidative stress from infection, certain drugs or even fava beans, hemoglobin oxidizes and forms Heinz bodies. These damage the RBC membrane, leading to premature destruction in the spleen or hemolysis. Clinically, this shows up as a sudden fatigue, jaundice, and dark urine. Interestingly, the genetic trait persists because of its partial protection against malaria. Now to summary and takeaways. So here are some review questions that I drew up. First, which of the following is the primary reason red blood cells rely entirely on glycolysis for ATP production? Second, the anion exchange protein known as Band 3 in RBC membranes is primarily responsible for. Third, which molecule decreases hemoglobin's affinity for oxygen, promoting oxygen release in tissues? 4, a patient with glucose 6-phosphate dehydrogenase deficiency is at increased risk for hemolysis because they cannot what? And 5, in the ABO blood group system, individuals with type O blood, what? And the answers are located on the last slide. So now to our takeaways. So to wrap this up, red blood cells are uniquely designed to transport gases efficiently from their bioconcave shape to their hemoglobin-packed interior. Their lack of organelles make them dependent on glycolysis, but also free space for oxygen transport. Their understanding their membrane structure, metabolism, and life cycle not only explains normal physiology, but also the mechanisms behind diseases like anemia, sickle cell disease, and G6PD deficiency. So final notes, just understanding the red blood cell biochemistry connects molecular details with real-world diseases like anemia and the ones I listed prior. So, thank you for your attention, and I hope you now see how much biochemistry and clinical medicine intersect the human red blood cell. And here are our references, the textbook of Chapter 53, and the photos were received from Google Images, and the answers to your review questions that I had listed before. Number 1 is B, number 2 is B, 3 and 4 are both C, and 5 is A. So thank you again for listening and on red blood cells.